evening, everybody. Today I'm going to present my research work that I've been doing in Dr. Raspalli's group for a while now. I've been involved in photosynthesis of hydrosins. My part was the synthesis of aromatic core. I'll tell you what all this entitles to, but first of all I will introduce, this is our outline of my talk. First I'll introduce hydrosins and then I'll show you how the hydrosins are made by nature and in our retrosynthesis. Then I will show you the plan for synthesis of my aromatic core, and then we'll discuss about some of the results that we have, and then finally conclusions and acknowledgements. What are hydrosins? Hydrosins are aromatic core containing polypeptide natural products. Again, I will show you the biosynthesis of this polypeptide pathway, how the nature makes this. But back in 2011 or 12, when we started this project, there were only 2 hydrosins that were isolated, and the isolation paper was published in 2005, and there were no synthetic studies available for this molecule, and they had some interesting biological properties such as antibacterial properties, and that got us started with synthesis of these compounds because nature doesn't make them in large amounts. So we had to we thought of developing the synthesis, and we thought of exploring the antibacterial properties for other bacteria as well. And when we were doing this, there were a few more natural products that got isolated in 2013, and that is more water under the bridge. That means there are more molecules that you can think of making. And though they all look a little dissimilar to each other when you look at the aromatic core, but they are all same when you look at the bridge. This is called ANSA bridge, and these are called ANSA aromatic cores, and these molecules are called ANSA lactams or ANSA macrolides or ANSA mycins, because they belong to a family of these ANSA mycins. So okay, ANSA mycins have a characteristic shape to them. There is something like a basket structure on the aromatic core, and there is a handle of a basket. That's why these are called ANSA mycins. So hydrosins are isolated from Streptomyces hydroscopicus, and that bacteria is well known to produce a lot of medicines that are already available in the clinic. For example, deltamycin is good for cancer. Some of these are antibacterial compounds, and there is a lot of need for antibacterial compounds. So I said one of the reasons why we are into this molecule, because we can develop new antibiotics, new molecules. Each time this bacteria develops drug resistance, we need to have a new drug. The drug is only good for so much time. After a while, the drug goes out of the market because the bacteria are no longer responsible, and there are a lot of these bad bugs evolving, and this infection society disease has come up with a research initiative where we are trying to come up with 10 drugs by 2020. That's the projection that they have, but so far we only have 2 drugs, so there's need for a lot of new antibiotics. How do these bacteria develop drug resistance? They develop with a lot of mechanisms. Though there were a lot of natural products which were drugs last 50 years or so, all these molecules are going out of the clinic one by one. The beta-lactams used to be very powerful drugs for bacteria, antibiotic drugs, but now they are not as powerful as they were because the bacteria is getting clever and clever. A lot of varieties of bacteria have developed a lot of resistance, and these are called bad bugs because they don't have any drugs to treat them. And if one would imagine that industry is aggressively trying to develop antibiotics, but opposite to that, industry is pulling out. In fact, you see in 1990 there were 18 industries that were doing antibacterial research. Now in 2010, this was taken in 2010, by 2014 there are only 2 or 3 companies that are doing the antibacterial research. This is contrary to the expectations. You would expect biopharma to be doing more research, but it's contrary to what we expect. The anti-drug resistance is going high. The industries are pulling out of this drug discovery research. They want to develop drugs for other diseases which are more profitable and fashionable, but antibiotics are not as fashionable as profitable to the industry. As a result, we only have 2 drugs that have been approved for the last 6-7 years. This was the data in 2012. We only have 2 new drugs that can be used for antibacteria, and by 2020 we need to have 10 of them at least to combat this, cooperate with this drug resistance. If not, we would be back to the pre-discovery, pre-penicillin era, where only the difference would be the body would be on a different mode of transportation rather than cause and caught. Otherwise, drugs come from? They normally come from natural products, and that's where especially macrocyclic natural products are known for antibiotic properties, such as vancomycin, erythromycin, rifamycin. These are already in the market, they were in the market, and they all came from nature. So that's where macrocycles have advantages. Why are the advantages to compare to the small molecules? Small molecules are not known to be enough to have these protein-protein interactions. At the same time, biologics like such as antibodies and other things would be good, but they are difficult to produce, whereas macrocycles have the advantage of the large molecules as well as they are better than small molecules. So this is some area between the small molecules and the biological area, biological products. So macrocycles are sought after in drug discovery. So I'm not going to go into the details. If you are interested, please go through this reference. You can read about the role of macrocycles in drug discovery. So now coming back to hygroxins, that was just an introduction to what we are doing. The hygroxins, as I said, there are 4 or 5 of them. Isolated from Streptomyces hygroscopicus. At this point, we wondered as to how to make them, and we looked at their biosynthesis. Nature is making, starting from this hydroxyaminobenzoic acid through a series of steps where polyketones are being built, and this polyketones chain has been built into this up to this aromatic ring, and this aromatic ring undergoes cyclizations, and we call this a biomimetic approach. This is how the nature is making. This is called polyketone pathway, and this is one of the approach in our lab, but I was involved in non-biomimetic synthesis. This is the non-biomimetic retrosynthesis, where I'm showing how we are planning to make in the lab. Instead of starting from one ring, we thought of disconnecting at this place. If we can make hydroxin A, we can make hydroxin B, because we just need to make one bond between this carbon and that carbon. That would be the 7-member ring. But if we disconnect at this point, this would become hydroxin A. So probably nature is making hydroxin A, and then hydroxin A is making hydroxin B. How can we make hydroxin A? Then to make hydroxin A, we can scissor this molecule retrosynthetically on the paper in 3 fashions. Then we can make this bond or this bond, and then we can disconnect this. This is an ester bond. We can always make an ester bond. This is an amide bond. That fragmentation will give us 3 fragments to worry about. Instead of one big fragment, now we cut the molecule, flat molecule to be made from 3 small fragments. Out of these fragments, my part was this aromatic core, and my next friend will speak about this part, and Gopi has been working on this chiral fragment. We all work as a team. Today I'm going to talk about aromatic core. So that's the target for my research. That has been the target for my research for the last couple of years, a couple of semesters, and I can divide this aromatic ring again for convenience sake into A and B rings, and we looked at the methodologies. The first approach that we thought was to go with the commercially available starting material that gives us all the substituents of the A ring, so we can buy this A ring, entire A ring along with the substituents, just 10 cents a gram. So what do we need to do here? We have to start from here and annulate the B ring so that we get the A B ring in place. Can we do that? That's what they thought in the beginning, and not going to go into details because some of the work was done before I joined the group. But this is how synthesis was done. Started off with an A ring, commercially available A ring. This is called salicylic acid, methyl salicylic acid. So through a couple of steps, we can make the amide, and then the formylation, and then protection of this phenol, and then the Wittig reaction, and then the peroxidation gives us this compound as a mixture of isomers. It can also go on this side, and once we have this compound, then the conversion of this carbomethoxy group was done through Cartesian rearrangement, converting to carboxylic acid and then treating with azide, gives us this amine, and amine was protected as Bob group. And if you look at the aromatic core that we need to have for the hydroxin A, this has all the pieces in place except for this oxygen. This oxygen is missing here, so the final step, however, has been the bottleneck, and it didn't work out as we planned. So we just published this part. The group has published this part in 2013, and we went back to the drawing board. This time we changed the approach. True, the A ring is commercially available. We wanted to go with the A ring, but the So we altered the approach. Instead of starting with the airing, we started with the bearing. Now can we start with the bearing and annulate the airing? This means this is more or less commercially available. You just have to annulate the airing through a Diels-Alder reaction. That's 4+2 cycloaddition approach. So this is the revised approach that I was planning. Just the sequence is different. Instead of starting with the airing, now I'm starting with the bearing. Let me show you how we did this. So the diene that is required for Diels-Alder reaction is being made in 3 steps, starting from commercially available solvent, which is some substrate, or you can also call it, this is like acetone, just metal substituted acetone. This is called ethyl methyl ketone, and that was treated with sodium peroxide, anion was generated and quenched with this formate that gives the formyl group, which is in the enol form here. The enol was alkylated to give this enol ether. This is enol alcohol, and this is an ether. So this is called enol ether. This is the enol form, enolate, and then methylation, enol ether, methyl ether is produced, and now we enolize this carbonyl, and then simultaneously we treat it with TMS triplet in presence of a base, so that protects the enol as TMS. Now this diene is locked in this form, so it can react with the dienophile. So this is the synthesis of diene, and let me show you how it started for the dienophile. This is commercially available. This is the bearing as I mentioned. Just it needs to be oxidized to quinone. This is an aromatic form. The aromatic form is not as reactive as the dienophile, so it has to be, this is electron rich. So we have to convert that into electron poor substrate. So we oxidize that to quinone using PIDA. The structure of the PIDA is shown here. This is the hypervalent iodine reagent. Iodine is hypervalent here. That oxidizes the phenols, the protected phenols into quinones. And this acetate was already commercially available, so we thought acetate would be a good protected group as well. So we bought it commercially available, so we bought it. However, the acetate is not as suitable as we thought. I will show you how it is not suitable in a minute. But having made this diene and dienophile, we subjected these two for Diels-Alder reaction, and the Diels-Alder reaction went very smoothly. It gave us this aromatized one. First it undergoes, first it forms double bond, but if you add silica gel to the same bond, it forms aromatic ring here and it forms a new bond, and it annulates the airing onto the bearing. So now we just need to functionalize this aromatic core so that we can attach other fragments. The functionalization requires the bromination on the airing, because this position is where the cyclone has to come to form the bridge. So we brominated it. However, the bromination was not very good, and also the deprotection of this acetyl was too problematic. So this is the crystal structure of that. As you can see, this is bromo. Thanks to Jim Bolen, we got the crystal structure and we know where the bromine is. Say there are 2 positions here. One could expect the bromine to go here, but this is more electron rich. So that's why that goes the auto-bromination than the meta-bromination, because this is auto-para-dielectric group. Right? So we have a quinone, we can use it, just that it's not very good yield and the protecting group is not easy to remove. So what we went back and we changed the protecting group a little bit, not completely changed. We just made this compound starting from aniline, treat with aniline, dimethoxyaniline with the BOC anhydride. This makes NH-BOC. We can make it broad scale and then we oxidize that again in PIDA. One can use PFOC. PFOC gives a clean reaction. So the quinone was ready, and then again we subjected to the same Diels-Alder reaction that we did before, only the protecting group was different, and it gave us as clean as a reaction of acetyl. So NH-BOC is now good because it's different than acetyl, it can be deprotected, and also we did the bromination. Bromination also works a little better than acetyl. Still there can be improved, but this is good enough to go forward. So we took this and we also manipulated the oxidation states again, because the quinones are not good for further coupling, and we'll show you the coupling. But for that we had to reduce this quinone again back to phenol and then alkylate in the same pot. This is sodium borohydride is a reducing agent, and methyl sulfate methylates the phenol, so we get the phenol back. So this is a reduction reaction. C13, and as you can see, there is no carbonyl anywhere. Everything is at this range, and I'm not going to go into the details, but this is where the aromatic water pattern lies. This is where the aliphatic carbons are, and you can see that there is a long carbon that is for the long group because you have 3 methyl groups there. Okay. And so let me summarize what I made. This is the whole chemistry. That is, we made the diene, we have the quinone to form 2 forms of protective groups, acetyl and log. Gives all the reaction and then reduction and then bromination. We have this one. Or one can do other way around. One can brominate it or reduce it. So nevertheless, we have this compound. And meanwhile, Gopi made this chiral fragment. This is a more demanding fragment. We have 3 chiral centers, 1, 2, 3, and this R group is methyl in my case. In another case, some other R groups, and when it's in the talk about neogenes. And nevertheless, Gopi has made the carbohydrate fragments. This is the structural group for the stereochemistry. He took my substrate and then he attached this substrate, and we combined to make this fragment, and we published this in Tetrahedron Letters in 2014. And this is the NMR for the complex intermediate, advanced intermediate that we made, and there is only one fragment missing. If you can close that ring, we have hydrogens, but we communicated this result in 2014, and we are working on the total synthesis. I am not going to go into the details. This is the compound we made, and see that there is a carbon here. That's for that carbon. This is for the solvent, acetone solvent. Okay, with that, I would like to thank people responsible for my training and my research in the group. And with that, thank you for your kind attention. Right? It's easy, right? You just don't have to worry about it. Just speak. Nobody is going to stop in between. Even if you say the wrong thing, you are not going to stop it, right? So then we'll stop and then discuss things. 17 minutes I spoke.